Hey everyone, in this video lecture, we are going to be covering the second half of protein synthesis, all about translation. So before we get into the actual steps involved in translation, just a little recap about the overall process of protein synthesis. So just remember that proteins are polymers. They are one of the major macromolecules that are necessary for life. And so your macromolecules are made up of a monomer polymer structure. So multiple monomers combine to form a larger molecule, the polymer. So in the case of proteins, the monomer is amino acids. There are many different amino acids, so the different combinations of amino acids come together. They're bonded together to create your polymer, which is called a polypeptide. It gets its name because the bond holding the amino acids together are peptide bonds. So there's many peptide bonds, hence we call it a polypeptide. So that larger structure, the polypeptide, is a protein, and we'll talk about what makes it a functional protein later on. So we've already talked about the first half of protein synthesis. It's a two-step process. So step one is transcription. We know that the DNA inside of a cell are the instructions for making proteins. However, DNA is way too large for it to leave the nucleus and get to ribosomes where proteins are created. So transcription starts by copying the DNA, or sections of the DNA, I should say, into RNA messages. And then what we're going to talk about today is translation, where that RNA message, which is really just a piece of copied DNA, will be translated into a functional protein. So in order for this to happen, we need to think about where in the cell translation is going to be occurring. So if we have this RNA message and we're trying to convert it into a sequence of amino acids, this must be taking place inside the ribosome. So the structure of a ribosome, the reason we call it a protein factory is it has these two subunits. The large subunit is the top half, the small subunit is the bottom half, and these two pieces of the ribosome are going to basically sit and wrap around a piece of mRNA, and the ribosome's job is to read the RNA and build that chain of amino acids. And these ribosomes are found both just free floating out in the cytoplasm or on the endoplasmic reticulum, which is why we have the rough endoplasmic reticulum. It looks rough because of all those ribosomes on top of it. So we're going to talk about in this lesson how ribosomes are protein factories, how they actually build that chain of amino acids by reading that RNA. And the way your ribosome will read the RNA are in sets of 3 nucleotides at a time. So we call a set of 3 nucleotides codons, because each codon, or set of 3 nucleotides in your mRNA, they will code for a specific amino acid. So we know that the codon, or the 3 nucleotides UCC, if they are there in your ribosome, the ribosome is going to add the amino acid serine, or SER. If we add the letters AUG, we know that the ribosome is going to add the amino acid methionine. So every set of 3 letters, every combination, every codon that exists has a known amino acid that would be coded for. And the way your ribosome is going to figure out, or the way that the ribosome knows which codon goes with which amino acid, is really this guy called a tRNA. So the T in tRNA stands for transfer. This is a type of RNA that basically will determine which codon goes with which amino acid. So it has this kind of clover-like structure, where on the bottom of it, it has an anticodon. This anticodon is complementary to the codon, meaning in your mRNA, whatever 3 letters are in the codon, the anticodon will match to it. They'll be the right complements to each other. So if there's an A, it would pair with a U, and so on. At the very top of the tRNA is a single amino acid. So this is what determines which amino acids get added. So how all this comes together is we have every combination of codon. So every possible 3 letters that could exist in an RNA, researchers have figured out the tRNA structures, so they know that if you have the codon UUU, that tRNA that would be complementary to that UUU would have the amino acid phenylalanine. Or, for example, if the codon is CCA of the RNA, they know that there's a tRNA with an anticodon that is complementary to that with the amino acid proline. So they've worked out every single combination of letters, which amino acid it would have. And so from this, we have this codon chart that would allow us to take the mRNA and predict which amino acids would be added based on those codons. So this gives us a lot of power to predict protein sequences if we only have the DNA or the RNA sequence. So how all this actually works inside of a cell or inside the ribosome. So step 1 for translation to occur, again, it's just like transcription, it's called initiation. So in order to start translation, what has to happen is the very first tRNA must arrive and bind to the RNA. And it will always, always, always bind to this start codon, AUG. This is always the signal for the start of translation. So the tRNA that has the complementary nucleotide, which AUG's complement is UAC, will come in and bind to the RNA. Attached to that tRNA is met or methionine, the start amino acid. And then once that attaches to the RNA, your ribosome will then come, both the large and small subunits will come and basically pancake around that tRNA and mRNA. So then after initiation, step 2 is elongation. So this is where the ribosome will basically move through the different codons of the RNA, and the complementary tRNAs will arrive into the ribosome, adding the correct amino acids. So, for example, in this piece of RNA, the next codon is UUU. The complementary tRNA would be AAA. On the other end of that tRNA is the phenylalanine amino acid. So once you have 2 tRNAs inside your ribosome, what happens is those amino acids are going to be attracted to each other and a peptide bond will form between the two. So then what will happen is the next tRNA for the next codon will enter into the ribosome. So if the next codon is CGA, the next tRNA is going to arrive that is complementary to that. That one has the amino acid arginine or ARG. So again, now that we have 2 tRNAs inside the ribosome at once, they'll be attracted to each other, form a peptide bond. The tRNA that is now empty, so that very first tRNA that arrived no longer has an amino acid together or on it. So that one will get kicked out of the ribosome. So at any point in time, there's potentially 3 tRNAs inside the ribosome. There's always that one in the middle where the amino acid chain is being built. There's one that is arriving and one that is leaving. And so the ribosome is just going to continually reading down each amino or each codon within that RNA strand. And then eventually, step 3 is we will reach termination. So the ribosome will be plugging along that RNA until it reaches what we call a stop codon. And there are 3 different stop codons. So in this example, we're looking at the codon UAG. It signals a stop. It means the ribosome is done translating and making that protein, and it's done. But there are no tRNAs that bond to stop codons. Instead, there's this release factor that comes in that will help the ribosome fall off the RNA and release your completed amino acid sequence. Now, once you have that chain of amino acids that has left the ribosome, meaning it's been created by the ribosome, after translation, that chain of amino acids is not an actual functional protein. What has to happen is that chain of amino acids or the polypeptide has to be folded on itself to actually become functional. And many proteins might have multiple subunits or multiple parts to it. So it might take multiple polypeptides to fold on itself to form one functional protein. So in this example, we're looking at the protein hemoglobin. It's a protein found in blood cells. So you have this chain of amino acids. It will form folds on itself, and then it will kind of form these sheets. And then there are multiple sections of these polypeptides that will come together. So in this one, there's like 4 subunits here to make the big structure of hemoglobin. So I recommend pausing the video here and practicing this how to translate. So you have a sequence of DNA, you have a sequence of RNA that is complementary to the DNA. So now what you need to do is use that codon chart a couple slides back and match each codon up with the correct protein or amino acid to figure out the chain of amino acids for this particular DNA sequence.